This Week in Parasitism is brought to you by the American Society for Microbiology at microbeworld.org/TWiP. This Week in Parasitism, episode number 38, recorded on April 2nd, 2012. Hi, everybody. I'm Vincent Racaniello, and with me is Dickson Despommier.
Hello, Vincent.
Good morning, Dickson.
Good morning, Vincent.
Actually, it's afternoon. It's 12 p.m.
It's just a little bit after 12.
Gorgeous day here in New York City, isn't it?
Perfect.
It is seasonal.
It is. Finally.
It's about 8 degrees centigrade.
Yeah.
Clear blue sky, sun.
No, not so clear.
Oh, it clouded up since I...
It's a Georgia O'Keeffe cloudy day.
Are you a fan of Georgia O'Keeffe?
I am.
Flowers and all that stuff.
Yeah, but she did clouds, too.
Did she?
Yeah. They're remarkable.
When I was making the agenda for today's episode, and I do for every podcast, I have an agenda.
You do.
Where we have notes about what we're going to talk about to keep us on topic. I guess it doesn't always work, right?
No, not necessarily, but
we have not recorded since February.
I know. It's not exactly this week in parasitism.
And here we made a New Year's resolution to do more.
We did.
So we'll have to try harder, Dickson.
I totally agree with you.
Should we change the name?
This weekend, this intermittent broadcast.
No, you could just call it Parasitism. Or Parasitism Now.
Parasitism Now.
Or Parasitism...
Up with Parasitism.
Well, give us some ideas for names that don't make it sound weekly.
Well, they want it to be weekly. We've heard from a lot of listeners that would like it to be weekly, and I know that there are other podcasts that adhere to that schedule without fail, but our schedule is such that it just doesn't work out that way sometimes.
Dickson is in huge demand.
It's not the idea.
You are in huge demand.
Well, if you say it that way, it sounds like I'm on an ego trip. But
No. I know that you want to feed people and all that.
Well,
I know you don't make a lot of money because your shoes are worn out and your pants are frayed. So.
That's a ruse. That's how I used to get a raise.
Did it work?
No, of course not.
So, and also in doing the agenda for today, Dickson, I realized we have a ton of back email.
Let's do some of those first then, Vince.
Would you like to?
Yeah, I would.
And if it ends up being an hour and a half, we can just call it quits.
C'est la vie.
All right. This could be the first all-TWiP email.
It could be.
All-email TWiP.
That's right.
The first one is from Carlos, who writes... I can't see this. This is too small. Let me make the screen bigger. As I age...
Oh, as we all age.
I can't see and I can't hear.
That's right. I'm sorry. What did you say?
I like wearing these headphones because I can hear.
Yeah.
Is there something equivalent for the eyes?
It's called glasses, Vince.
No, I have those.
Or lenses of various sorts.
Carlos writes, I am an avid listener of TWiP since its start. I have been following TWiP for at least 2 years, and surprise, also follow TWIM. My field is computer science, but I crave for information in all areas I find interesting. To me, your podcasts fill the role of giving me very pleasurable conversations on science and help me better understand fields where nature dictates the validation of the research. In my field, we often deal with the construction of artifacts that are validated by their mathematical properties. So the validation process is a bit more detached from nature.
Well, I don't know about that. It's an interesting observation. But if you look back at the history of mathematics, I'm not a mathematician, but I'm a fan of the history of mathematics. You can go back to this guy Mandelbrot, and he came up with this Mandelbrot set that looked totally artificial and generated figures that didn't have anything to do with nature. And the next thing you knew, we were applying fractal mathematicians' theorems to the way the heart beats and the way mountains are formed and the coastlines of countries. And it turns out everything is a fractal. And so fractals are, popped out of our head. So I'd love to know from this listener where he thinks mathematics actually comes from, because it's been a conundrum for me to imagine the origins of mathematics from your brain. It wasn't there before there were humans, although the constructs were. But where does mathematics come from? Where does it originate? Is it our ability to express the way we're constructed through matter? And that's very philosophical, of course.
I don't know. It depends how you view math. I mean, some people feel that it's the underlying truth to everything, but others feel that it's a way of expressing the world. But if you went to another planet, they would have a totally different math, right?
Not necessarily.
I don't know. That's a good question.
If nature is the same everywhere, and if the same 92 elements are used to construct their world as
that's chemistry.
Ours. Yeah, but that's math too, though.
Is it?
Yeah, sure. The atomic...
So you think math is truth?
I do believe that is right. That's right. So I'd just like to know, if you wake up one morning and you come up with this amazing mathematical solution to a previously unsolved problem, where did that come from? How did you actually do that? And you ask any mathematician, and they can't tell you. They just said, the idea occurred to me. But how does that idea occur?
Well, they have to prove it, though, and they use math for that.
Yeah, no,
right?
you're right. You're absolutely right. But still... I'm quite amazed at that because I have no mathematical abilities whatsoever, and so I'm in awe of people who do.
I'm going to give you a math quiz. Just
don't do that, Vince.
A squared plus B squared equals C squared.
Yes.
What is that?
That's the quadratic equation.
No.
See, I'm lost. I'm totally lost. A plus B equals C.
No. A squared plus B squared.
No, you're going to eliminate all this from our discussion.
Is the Pythagorean theorem.
The Pythagorean theorem.
The sides of a right triangle.
Yeah, I'm out of touch.
Anyway, he continues, what prompted me to write now is that I just listened to the start of TWiP 33 and your discussion of how Google Scholar indexes more publications and citations than PubMed, I believe, but with a more messy presentation format. You might find it useful to explore a new tool that allows authors to aggregate papers in a specific Google Scholar author page. After one does that, it's easy to sort by publication year, impact, etc. Take a look at the following link to see how to set it up. And he sends a link to Google Scholar Citations Open to All from November 2011. Okay, I will check that out. It looks good.
Yep.
The strong point is that the tool makes it very easy to identify and separate your own publications and co-authors. For more common author names, it's often a mess to separate one's articles from the other homonymous authors.
Huh.
You know that word, Dickson, homonymous?
I don't actually, Vince. What does it actually mean?
Well, a homonym, right?
Right.
It's two words having the same spelling, but different meanings and origins, like read and read.
Right. Or
right?
Table and table.
How about read and read? That would be better.
That's right.
Table and table. Keep doing the excellent job of promoting interest in your fields and science in general. Carlos is from the Universidade do Minho in Portugal. It's our second Portuguese listener, I think.
Thank you for listening, by the
We
way.
have another in Roberto. All right, the next one is from Peter, who writes, just watching a clip on cellular metabolism. The background music was from the Vangelis theme from Carl Sagan's Cosmos. A very great sadness came over me. He looked outward, whilst you guys look inward, so to speak. I feel sure he would have loved your shows and would eagerly have embraced podcasting. You are doing the inner cosmos. Thank you.
That's wonderful. That's a great comment.
What does that mean, Dickson?
Well, I think it means that we are not afraid to reveal scientific truths.
As far as we know them.
As we know them. And then when we stumble every now and then, which we do, everybody does, then we get these wonderful emails from people that correct that. So I love the fact that there's no ego tripping involved in any of this. I really do.
And someone over on TWiP wrote that they also like that we admit when we make mistakes, which he said is not always the case, especially in an election year. Dickson, I think Carl Sagan would have liked podcasting, but he was bigger than pod. I mean, he had radio shows, TV shows, right?
Absolutely.
He didn't have to meddle with this.
No.
I think he was too famous to...
But today, in today's world, actually, he would have had huge audiences if he had podcasts.
Oh, he would probably have his radio show distributed as a podcast. Did
Sure.
he have a radio show? I don't even know
I
if he did.
don't recall.
He could have done pod, but then he might have. How did he talk? He would have said, "I don't have time for that. I'm on TV."
No, he would have said, "We have billions and billions of listeners."
That's right.
He was very dramatic in his approach, but also very provocative.
Who is the Carl Sagan of virology?
Got to be Vincent.
No.
Racaniello.
Is there one? No.
A
one has-
provocateur,
no
a provocateur.
one- just someone who the public sees and learns from. So that was
Tony
his role,
Fauci.
right?
Tony Fauci probably.
He
was
doesn't
the-
teach, though.
No, but he does. He's on television quite
Let
a
me
bit.
say right here and now, I want to be the Carl Sagan of virology.
And you both went to Cornell, so you could, this could
Yeah.
happen. This
I
could
don't have
happen.
his personality or his knowledge and
You
intelligence.
don't know,
but
Vince. You have your own style, and I think it plays very well on these formats.
The next one is from Emerson.
Okay.
Hi, guys. Love the show. You listed CSF, blood, lymph, urine, aqueous humor, cochlear fluid, and semen. You remember, Dickson?
I do. Those were the 7 biological fluids that were sterile.
Now, he says, I'd like to suggest that there are more than 7. Ignoring interstitial fluid and cytosol, I'm fairly sure that bile is sterile before it leaves the gallbladder. Synovial fluid within the joints is typically sterile. The fallopian tube's ovaries are bathed in sterile fluid. Tears are also sterile before they are cried, I believe. It's great. I love it. Before they are cried. Wonderful. Take care. It's probably right, no?
I would agree with all of those.
Why do you say there's 7 then, only, Dickson?
Well, when I was in school, that's what they emphasized.
Oh,
They
no wonder.
majored the
This is
7.
like 100 years ago.
Not quite so long ago, but I mean, those were the major ones that they used to list. But synovial fluids, of course, that's sterile fluid as well.
Okay.
Bile, I'm pretty sure is sterile, but not 100% sure.
Do you know the 7 non-sterile fluids? I don't. All right, he's right.
Yep.
And there are probably more, if anyone can
And
think.
there's probably more. That's right.
Laura writes, I'm a master's student doing research on soil microbes.
Uh-huh.
And in my research, have come across papers referencing molecules, phytoplasmas, microplasmas, ureaplasmas, and spiroplasmas are example. Are there any parasitic molecules that actively infect humans and cause detrimental symptoms? Do you know this phrase, molecules?
I'm not familiar with it. Sorry.
Mollicute.
I am not familiar.
Class of bacteria distinguished by the absence of a cell wall.
Really? I mean, like chlamydia or something like this.
Yeah, mycoplasma, the best known
Okay.
is mycoplasma.
We call them other things in the infectious disease world. Molecules.
So these are parasitic because they need to live in the cytoplasm, right?
Yeah.
So she wants to know if any are any parasitic actively infect people and cause detrimental symptoms.
That's a good question.
Well, the mycoplasma do, don't
yeah,
they?
they do. Yep.
Which is one of the ones that she stated herself, in fact. Spiroplasma, mycoplasma. So this is a question for TWiP.
It is. It sounds like more directed towards prokaryotes than eukaryotes.
Rickettsia, do they have cell walls? I believe they do.
They do.
All right.
They fall just short of a complete bacterium.
All right, so we will toss this over to TWiP, but I'll bet we have some listeners who can answer it.
You know, speaking of Carl Sagan, remember he was married to Lynn Margulis.
Yeah, who died
Yes, unfortunately.
recently,
Or I
last
guess-
year.
Yeah.
Mm-hmm.
Yeah. So, but she was really careful about trying not to make too many distinctions between life forms, because there are gradations of life forms that go from the infinitesimal up to us.
Mm-hmm.
And if you start arranging them in order of complexity, you might get a surprise or two based on something that leaps ahead of something else that you thought should have been ahead of it based on other characteristics.
Mm-hmm.
That's to simplify it, I should say this. So remember, we've had this controversy in TWiP as to what constitutes life. Remember we talked about this a
where
lot.
the viruses are living, which
Yeah.
they clearly are not.
Well, we don't think so because I think the concept of metabolism enters into the picture as part of the definition of life. And viruses have no innate metabolism of their own, whereas
That's
everything
true.
above that does, including chlamydia and things like that. So, it's too bad we didn't think of that argument when we were discussing it, but it just occurred to me recently
I
that,
think that was brought up.
I don't recall.
It depends if you. A lot of people don't use that as a criteria for life.
I know they don't, but I think they're wrong.
That's
Just
the
to
thing.
throw that out there.
It's not set in stone. It's not like mathematics.
No, it's not like, that's true. Isn't that interesting?
Some aspects of math.
Well, if
right?
mathematics reflects life, then that's set in stone too. Then somehow everything seems to be set in stone. Then,
I don't think
unless you
that's
get down
right.
to the quantum level,
How
and
you
then things get
interpret it
fuzzy.
is not set in stone.
The fuzzy levels of quantum mechanics.
So, getting back to Laura's question,
Yeah.
many molecules cause disease in humans, attaching to cells in the respiratory or urogenital tracts, and particularly Microplasma, which does respiratory pneumonia,
And
right?
chlamydia.
And ureaplasma, another one. So there you go. The answer is yes.
Right.
The next one is from Peter, who had an email previously about Carl Sagan.
Ah.
Taking the image challenge on the New England Journal of Medicine site, I guessed wrong on a lung sputum sample. Desperately trying to recall lung involvement parasite. Ascaris? Bzz. Of course not, dummy. Too quick on the mouse. It was a bloody egg in the lung, not a juvenile organism, and I don't think Ascaris does the disgusting stomach-to-lung thing.
It does, actually.
Turned out to be Paragonimus.
Aha.
Paragonomyiasis.
Uh-huh.
I don't recall that one in TWiP. Sounds interesting. Coming soon? Pause. Ignore last email. Duh, I realize you guys did touch on Paragonimus.
Yeah, we did.
TWiP 27.
That's right, we did.
Trematodes. Must have some in my brain.
Well, not necessarily. But Ascaris actually does have a cycle that goes through the lungs. But not as the egg. It's as a larva. So it has to actually go through the lungs.
So this is an image challenge from the New England Journal. Have you seen this?
I've seen some of them.
Would you like to be challenged?
I would love to be challenged.
If I can turn this without disconnecting our recording here.
Right.
All right, so that's the first one there.
Okay.
Do you know what that is?
It has a smooth edge to it, so it looks like a collapsed something. Is that what is the sample?
Yeah, so there's no labeling here, right?
It doesn't show you the label.
There's 335 images. Wow.
Well, we could go on and on about this one.
Here's a picture of a lung. Let's see this finger, this badly swollen finger here.
Yeah.
I don't know what that is.
That could be a spider bite.
It looks like there's something solid in there.
It does. You know what? That could also be Paragonus. I'm not joking. Sometimes that adult worm can migrate into some aberrant spots, including the finger or your brain.
Anyway, we'll post this. This is pretty cool. Thanks, Peter.
Very nice.
335. You could spend hours doing that.
You're right. I used to be part of a group for the American Society for Tropical Medicine and Hygiene in which they sent out slides, actual microscope slides with stuff on it. And they gave it as a quiz to anybody that was doing diagnostic work. Mm-hmm. And then at the meeting, they would announce the winners. And I used to be good at this. I've lost my touch.
Didn't you tell me you took all the photos in your parasitology
I
book?
took all the photos. But the one thing I didn't take a photograph of, which I should have, was something called respiratory pneumonia. Not respiratory pneumonia. That's not the right. It's when patients regurgitate food and then inhale it. So it's called inhalation pneumonia. That's the term, I think. And the starch granules that are left over from meals that contain starch in them, like, say, pasta meals and stuff, those little granules of starch grains look exactly like some of these pathogens in terms of their eggs. So when you do a cross-section on it and you think you're looking at an egg, you're really looking at a starch granule.
Interesting.
Yeah,
some famous rock stars died that way.
Yes, I do.
They vomited and inhaled their vomit.
Mama Cass died that way.
Who else?
Oh, that's a good question.
Jimi Hendrix.
Did he really? I didn't
Janis
know that.
Joplin.
I thought she died of an overdose of heroin.
I think they all end up asphyxiating on their vomitus.
I see.
The next one is from Brian.
Yes.
Doc's R&D. Ooh, do we do R&D?
R&D. Hey, we never even thought of
I
that.
haven't either. At the end of your Alum Recordes program, you discussed problems related to Toxocaracanus and T. cati infection in humans, namely VLM and OLM.
Correct.
Dick said it would be great if someone surveyed dog runs in New York City or nearby for Toxocara eggs.
Right.
Sampling and counting eggs sounded relatively straightforward. Piece of cake was mentioned a couple of times. Such a survey would be an excellent topic for an enterprising high school student with some help from a famous parasitologist or virologist or someone they may know. Give it some thought. Didn't our friend in Brooklyn isn't he involved in that?
There is somebody in Brooklyn.
Michael Walsh, right?
That's correct.
We had him on TWiP, do you remember?
We did. Yeah, I do, of course.
So he's done that sort of thing.
That's right.
They get dirt samples from various parts of the city.
But they're not looking at dog runs.
Actually,
no.
they take feces, right?
They do.
So remind me, do you remember if Michael was looking at dog runs? I don't think so. Right?
No, but I suggested to him that he should include that into his survey.
What did he say?
He said it was a good idea.
So there's an episode of TWiP where we talked with Michael Walsh, an epidemiologist from Brooklyn.
That's right.
And he is trying to get funding to continue this work of looking at fecal samples on the streets of New York, right?
On the streets of New York.
And then he counts...
And in the parks.
Eggs, is that right? He looks at eggs?
Yeah. The eggs are resistant to lots of things, so they'll last a long time.
I'm reading Dick's book pick a while back, The Fever by Shah. I find it fascinating. Do you remember that one?
I do.
As an anthropologist, I am well acquainted with the sickle trait balanced polymorphism, but underappreciated the impact malaria had on human history.
Oh, wow.
The rapid spread of the parasite in the U.S. hit home after reading how fast it infected areas of New England after the environment was altered by the building of mill ponds. It took only a few returning soldiers from the Civil War to infect whole towns. Incredible.
Here.
Mill ponds?
Yeah.
To grind wheat?
Well, you build the pond above by damming up the river, and then the water that spills over the dam drives the wheel that grinds the whatever you're grinding.
With the grain, right?
The grain in some cases, but they also used it for power to make shoes, leather, stuff like that, furniture.
And this allowed mosquitoes to breed?
Yep. Instead of a running situation, you had a standing situation.
Wow.
Yeah.
My wife took a parasitology class a few years ago and had to draw many of the specimens you have discussed. She thought Giardia trophozoites were cute.
They're fun.
Little faces. After going through a Giardia infection, I'll tell you how cute they are.
Yeah, right. Exactly right. Cute, they're not.
Have you ever had Giardia, Dickson?
No, I've never had Giardia.
As much as I love TWiP, I am happy with the longer break between episodes, so I have time for the many other terrific podcasts like TWiM, Science Friday Video, a favorite if you haven't seen it, New York Times, The Minimalist, Science Now, Nature Archaeology Channel, Radio Lab, among others. So many podcasts, so little time.
Wow. And just 2 ears.
Huh. That's a lot of podcasts.
If we had more ears, we could listen more.
And I have to know how you selected the fun but creepy opening music and great hard-driving riffs at the close of the show. Good stuff. Cheers, Brian.
Thanks, Brian.
The music was selected. All right, so it's performed by a fellow named Ronald Jenkins. There's actually a link to Ronald. I think it's ronjenkins.com. Let's see. Nope. ronaldjenkins.com. Let me just search for it. Stop looking at your watch, Dickson.
No, I'm just gauging our discussion because I'd like to include a parasite into this issue, that's all.
Yeah, we'll just do a bit more. What's his website? ronaldjenkins.com. It's all one word. My son likes his stuff, and when I was looking for music for TWiP, he said, Dad, you should look at this. So I listened to a few, and I liked them, and I emailed Ron. I said, can I use this? He said, absolutely. I love educational podcasts.
Oh, great.
And in fact, TWiM music is also by Ron. And the only one that isn't is TWiV, which is just stock music. But I think I should change it.
Yeah.
Because it turns out that other podcasts use the TWiV music, and that really bothers me.
Oh.
And I want to get something unique.
Maybe some of our listeners who play Moog synthesizers will send us some of their original compositions.
You know, in TWiV, we've had a listener draw caricatures of us.
I remember.
If we have a musical listener, maybe they could make something.
Sure, we'd be glad to use it, by the way.
The next one is from Trudy. Listening to you right now on Science 360 Radio, which, by the way, is an NSF-run website where they collect science podcasts, and all ours are there.
Nice.
I wrote a grant to the NSF.
I recall.
This, but
hopefully they'll hear this podcast and respond positively.
You mentioned this post, and I had to comment. As a former nurse epidemiologist who worked in county public health practice, I am a strong proponent for vaccination. I worked a number of vaccine-preventable disease outbreaks, including measles, which resulted in individuals becoming needlessly seriously ill. Very much enjoyed TWiP, and TWiM. My mother was also a nurse who worked in public health before vaccines were available. She saw the tragic cases of diseases which resulted in great morbidity and mortality. I could go on and on about the good vaccines do. Thank you. Kudos to you and all who assist in the production of wonderful podcasts. Dr. Despommier is a treat.
Thank you.
I have so many interesting experiences with all kinds of reportable diseases and conditions, including how I became a nurse instead of a medical illustrator or possibly a physician. I sustained a needle stick pre-recombinant HBV vaccine while working my way through college in a teaching hospital. I carry natural immunity to hepatitis B now. Didn't get any extra credit in my micro class, however. Trudy is from Naples, Florida. Maybe you would like to come and tell us about your experiences sometime.
Yeah, that would be interesting.
The next one is from Ricardo, our friend in Portugal, our other friend in Portugal. Hello, Vincent and Dickson. I just came from my first jury in a monographic work where the student cited TWiP. The monograph title was Trichomonas from the Parasite to the Ecosystem.
Oh, wow.
I was very impressed with the watermark left by TWiP on the student, and happy that my friend, her supervisor, suggested her to listen to TWiP.
Wow.
One of my colleagues from the jury commented, the text looked like a romance. She appreciated the stories. Thank you both for the really great work you are doing. So
Thank you for
Ricardo
listening.
is a professor of microbiology at the Fernando Pessoa University in Portugal.
Nice.
Felix writes, Hi, professors. In episode 16 on Giardia, you discuss Antony van Leeuwenhoek. Is that right?
Yes.
And his early adventures with a microscope. Here are a couple of interesting resources which discuss him.
Oh,
Firstly, an article in ASM magazine claiming that Leeuwenhoek gets too much credit for his discoveries, some of which should be shared by Robert Hooke of the Royal Society of London. That's a link to that. I have to tell you, I'm reading a book called Quicksilver right now by Neil Stephenson, and Robert Hooke plays a major role. It's fiction, but it's his interpretation of the era of Hooke and Newton. And all this.
Here's a pop quiz for you then, Vince. Who did Robert Hooke marry?
I don't know.
He married a woman who was involved in the French Revolution, who was married to Lavoisier, and Lavoisier was executed, but before he was executed, she escaped to England and became the wife of Robert Hooke.
Oh, I didn't
So
know
it was
that.
Lavoisier's former wife. Lavoisier, of course, as every Frenchman knows, lost his head over science, as it were.
Robert Hooke is quite a character, according to this book.
Exactly.
Secondly, an excellent BBC documentary, The Cell, which begins with Hooke and Leeuwenhoek. You have also mentioned Paul de Kreef's book, The Microbe Hunters, as one of the few sources for information on the history of germ theory and microbiology. A recently published book, also from the ASM, addresses the same area, most likely with a less idiosyncratic style than the dated de Kreef volume. It's called Germ Theory, Medical Pioneers in Infectious Diseases by Robert Gaines. Just while writing this email, I discovered the ASM bookstore with a section of the history of science. I have no connection with ASM. I'm currently re-listening to all of your TWiP podcasts, having listened to them all at least twice when they were first released. They are just that good. Thank you so much.
Wow.
I really love the beginner-level approachability of the first 30 or so episodes. It would be great if you could cover a small number of highly important non-parasitic diseases such as cholera in the same style.
TWiM.
Yeah, we just don't have the Dickson Vincent thing on TWiM. Not yet, anyway.
No, not yet, anyway.
There's nothing, Dickson. There's just not another person like you in microbes.
Do
you know a microbe person that is like you that could have this
Well,
sort of repartee?
I've heard Rita Caldwell many times, and she's a delight, and I would highly recommend getting her on TWiP.
The cool thing about TWiP, Dickson, is that you and I are always on it.
We are, but we're colleagues. We're right next door to each other.
So you have a constancy
that's
based
true.
on that. But we don't know all about these bacterial infections, so we would have to get
know
someone.
about some of them. We know about some of
I
them.
know, but you know about all the parasitic
The
infections.
cholera,
That's amazing.
I could probably do a decent job of an elementary expose of cholera based on my medical ecology website, which I had to actually go into really details about how
All right,
cholera...
So I need to do a podcast on, it's like a TWiP of bacteria.
Yeah.
TWiB.
TWiB, that's right.
And get one person who can do the repartee.
Yeah, that's tough.
The next one is from Jim. Hi, I'd really like to hear the details of your efforts to convert Dickson's text to electronic format for access over the web.
Right.
Lessons learned should be useful to anyone with similar thoughts.
Hear, hear.
How is it different from creating such a work from raw materials? I
Oh.
prefer it as part of the TWiPs if that is accessible.
Yeah.
I still hear all of the TWiM, TWiP, and TWiVs, but my podcast listening load is heavy, 60 per week.
Oof.
And production of a weekly blog about the best entries is slow for an old-timer. Love Dickson's link to the stop-action high-speed photos of the liquid drops. Remember that?
Yeah, I
Dickson?
do. I sure do. Sure.
Our efforts to convert Dickson's text. Well, so Dickson has the text in InDesign, which is a page layout program by Adobe.
Right.
And so this was originally in 2-column format.
Yep.
So I went through and converted it to 1 column and had to do it manually. It took, I would say, about 12 hours, right?
Long time.
And then there is a plugin for InDesign that exports to Kindle format. So we actually ran it over the weekend because we started it on Friday.
Right.
And it was taking hours and hours. It just finished this morning, I think. And we looked at it, and there are some issues. I mean, there are formatting issues. The figure legends are all over the place. They're not with the figures. The figures are just moving around randomly, and we don't have a table of contents, which we have to do.
Or an index.
And there's no index, so we're not done yet.
No. But we're heading in the right direction. And by the way, I must say, Vince, that the pictures on Kindle look so much better than they do in the book because they are the original scanned pictures. Right. There's no loss of detail as the result of the printing process. And I'm excited to see what's going to happen when the iBook version comes and you can actually expand each figure and make it fill up the entire page. I would love to see what that looks like. So I think what we probably will end up doing is making page breaks between parasites so that each one will have its own unique set of pages.
That's a good idea.
Somehow we can add some more spaces and like this.
And then what we so the Kindle experience is not good because the final view is very small. The Kindles are rather small and it's not perfect. So we're going to do it also in the Apple iBook textbook format.
That's right.
And you've written already a book in that format, so you find it easy, don't you, to put text and pictures together?
I have, yes, and it does go together much better than if you started with a book that already existed.
Right.
So starting from scratch is the best way to start all these things, I think.
So what we'll do is copy the text and paste it and then put the figures in individually. But either way, the layout, making a nice layout is the key so it looks good.
That's right.
And that's hard.
It is. iBooks allows you to make the layout as you go along, by the way.
Yeah,
as long as you do it in the landscape format.
Yeah.
If you do it in the portrait format, then it puts the things on a little, like thumbnails along the side. Then you have to find them in the text, and that's more difficult.
So we'll keep working on the Kindle until we get it right, and then we'll upload it. And
exactly.
We'll let you know how that
Shouldn't
goes.
be long.
And then when we do the iBook textbook, we'll let you know.
Right.
This is from Sven Urban, who is from Sweden and has written us on TWiP before. Dear Professors Raqueniello and Despommier, as a longtime listener of TWiP, and for that matter TWiV and TWiM, I feel obliged as well as entitled to express some mild criticism that I feel and hope is appropriate. While your mutual friendship is a much valued aspect of the show, I think it might lead you to a degree of bantering that interferes with the contents. Slightly too often I find your discussion interrupted by mutual jibes, which makes me lose the thread of thought. I would say that generally, though not exclusively, Dr. Raqueniello is the instigator of these interruptions. I am, of course, not now referring to those abundant, seriously intended, and equally seriously phrased questions that Dr. Racaniello interjects, but rather about occasional stray comments and jibes that appear in private between very close friends. I think the term in private here is crucial, as I doubt that you would express yourselves like this, were the two of you jointly presenting before a class. Today, I could, by the way, compare TWiP 37 dracunculiasis with TWiV 172 with Kathy Spindler, both in a one-on-one setting, and the difference in tone is remarkable. Naturally, I speak here only for myself, of course, but I, for one, would appreciate it if you might henceforth tone down the sparring, keeping in mind that you actually do appear before a large international audience, not all of whom might appreciate or be able to follow your bantering. And just to be precise, tone down does not necessarily mean discard. This notwithstanding, I am very grateful for the set of podcasts you so graciously provide us all with. Sincerely and respectfully yours. Absolutely.
That was very carefully worded.
I got this a month or two ago, and I said immediately, you're right. I'm sorry.
Well,
I got carried away.
Because we're just sitting here in New York City and we happen to share office space with each other, we're adjacent to each other, and we see each other every day, it's hard not to behave casually for a podcast because it just sounds like we're sitting down and having a conversation, but he's absolutely right in the sense that we don't know how many other people are out there listening.
Well,
right?
I do, and I appreciate all of them, and I don't want that to interfere with the teaching because that's the most important.
No, we quite agree with that. So where is...
You know what I do? I'm sorry, what?
Where in Sweden is he?
I don't know. I'm sure he would let us know. Before, I know you're going to talk about going to Sweden to build a vertical farm, right?
Correct.
Before you do that, when I teach virology, Saul Silverstein is sitting in the back.
Yes.
And once or twice in a lecture, we'll banter. I'll say, Saul, what do you know about this? But much less than we do. So criticism is absolutely appropriate. I will tone it down. And if you notice today, I toned it down a lot already because I knew this email. So Sweden, what were you going to say?
Well, if this gentleman is anywhere near Linköping, which is in the southern part of Sweden near Göttingen, then there's a company called Plantagon, which is in construction as we speak, making a 17-story vertical farm for this small city. Not as a demonstration project. This is a real vertical farm that will be commercially successful because they've already selected their crops based on consumer demand, and they know they can produce them inside. So I think this is a very exciting turn of events for the concept.
Are you going to visit the farm?
Eventually, yes.
Well, you should hook up with Sven Urban.
Well, that's why I asked you where he was, because maybe I will.
The next one is from Robin. Actually, he has two. He provides us a definition from the freedictionary.com of acta. I think we were discussing acta.
Yeah, we were. Yes, we were.
Often, acta, official records as of acts, deeds, proceedings, transaction, or the like. Origin is Latin. It is the plural of actus, agere, to do, cf. act. Okay, thank you. And Robin also writes, the TWiP podcasts again emphasize to me the difference between hearing firsthand from someone with experience in contrast to hearing material regurgitated by someone with book knowledge. It made a world of difference listening to a trauma surgeon or an emergency physician from a trauma center speak in contrast to similar material presented by someone without direct experience. Mm-hmm. This is my philosophy of scientists teaching science. We have the direct experience. There's nothing that can come close.
No, that's true.
Sarah writes, greetings, Dickson and Vincent. I am a new listener, and I am thoroughly enjoying working my way through the TWiP and TWiM podcasts. I found your conversation regarding dracunculiasis very informative. I think it's great that you spent a good portion of time emphasizing the necessity for clean water for overall public health. It is something that we often take for granted here in the U.S. Something caught my attention, though. You talked about the use of cheap cloth to filter water and the prevention of guinea worm, and mentioned that it was unfortunate that this wouldn't prevent diarrheal diseases.
Right.
I attended a session at the ASM General Meeting a few years ago to the contrary. While sari cloth won't prevent viral-borne diseases, it does seem to reduce the incidence of cholera. And here is a link to the study, which was published in the first issue of MBio. Sarah is a professor of biology at Gordon College. Do you know where that is?
I don't.
It's in the state of Georgia. This MBio paper, simple sari cloth filtration of water, is sustainable and continues to protect villagers from cholera in Bangladesh. And who do you think is the last author on this paper?
Rita Caldwell.
Yes. How about that? This is pretty cool.
I think that's great then. I am happy to retract my statement then. More than happy. I was totally unaware of this subject. But I was thinking of things such as cysts for Giardia and histolytica, for instance, which might be trapped. I mean, they're a little bit larger than the cholera organism. But remember, cholera has this very long flagellum, and maybe it gets all tangled up in the microfibers of the cloth.
We reduced cholera incidence in this area of Bangladesh by 48%.
You know, something like that in Haiti would go a long way too, wouldn't it?
If you got rid of the UN people, it would help.
Yeah, well, it's too late for that because it's now ensconced in the general population, and they're but dreading another outbreak.
This is funny. This next one, Ian writes, Dear Doctors Raqueniello and Despommier.
Okay.
You changed your name.
We're going to try not to laugh at these things because we don't want to get too...
No, you don't have to go too far. You can laugh, Dickson.
Oh, okay.
It's my fault. I do too many jibings of you.
It's not your fault.
I'm immature sometimes.
Stop it.
In your presence, in particular. I don't know. You bring something out of me. On the topic of the dracunculus life cycle, is there evidence that a male is required for fertilization?
Sure there is. Yes, of course. I mean, is there evidence for it, or is it just assumed?
That's asking if there's evidence.
Well, I have not seen anyone who's shown a picture of the male dracunculus organism, to be honest. I have not. It's always assumed to be a diminutive form, and then after mating, it is assumed to have died and gotten eaten up, through a granulomatous reaction. But I have never seen a picture of one, and I have never seen... the only evidence that exists is the fact that it's a diploid organism, and that you can probably see the sperm from the mating in the oviduct of the female. So that you know somehow that occurred, but where and when is up for grabs.
As he continues, some nematodes are parthenogenic.
Yes.
And virgin birth for this worm would be a nice complement to the biblical story of the caduceus.
That's absolutely true. I mean, Strongyloides comes to mind here too, so you could have two of these, but they're totally unrelated worms in that sense, and none of the filaria that I'm aware of have ever been implicated in terms of that.
Okay. Alicia writes, hello, I've heard that Toxoplasma gondii might be caught by touching a leech. What's the general consensus on this?
Touching a leech?
Yep.
I've never heard that one, but that doesn't mean it doesn't exist. It just means I haven't heard of it. Touching, that sounds pretty spurious.
Does Toxoplasma infest leeches?
No, but I mean, I think if leeches were to... Well, a leech is a blood-sucking organism, first of all. Okay, so it's an ectoparasite, it's considered to be. Toxoplasma, on the other hand, all right, Toxoplasma gondii. It's tough. It's very interesting to speculate about it.
Well, here's a paper from 1994 in Parasitological Research. You know that journal?
I do.
It's a top-tier journal?
Compared to what? Science or Nature? No, I wouldn't call it that.
Experiments on the possible role of leeches as vectors of animal and human pathogens, a light and electron microscopy study.
No, some leeches are known to transmit trypanosomiasis, for instance. So it's well established that leeches could transmit a disease. When just touching a leech, I think that doesn't mean you've been bitten by a leech.
Yeah, it says here that they observed the reproduction of toxo in the gut of the leech.
Reproduction?
It said here, it says we're even capable of reproducing inside the gut of the leech.
Really? Mm-hmm. I had not known that. But this is such a generalist parasite that I'm not totally surprised by that, I guess.
The problem with touching is that it's in Wikipedia.
Oh.
And it says, ingestion of contaminated cat feces, but it says this can occur through hand-to-mouth contact following gardening, cleaning a cat's litter box, contact with children's sand pits, or touching a leech.
I would question that one.
Is the leech contaminated by cat feces? Is that the idea?
No. Leeches are found in the mucky bottoms of ponds and lakes, that sort of thing.
Right, so your answer is no. You don't know of anything.
I don't know of any.
All right. You want to continue, or do you want to move on to I have many more, but...
You do. I would rather do a parasite now. I think we should switch to a parasite.
Okay. Well, we've been doing parasites.
No, but I mean a new parasite that we haven't done yet.
I think that's a good idea.
Right.
What would you like to talk about?
Well, since you revealed something of your past to me during an interlude before we started, I'm compelled to, on air, find out exactly what your role was with regards to biomedical research of Trichomonas vaginalis, one of the most commonly occurring sexually transmitted, I wouldn't call them diseases all the time, but they're certainly a sexual infection.
Mm-hmm.
It's a protozoan. I worked for 3 years at Rockefeller with a wonderful gentleman by the name of Miklos Mueller, who became an expert at this organism and worked on its cell biology. So I'm familiar a little bit with some of the early findings with regards to trichomoniasis. And in fact, I was the one that, through a resident here at Columbia's OBGYN department, identified the very first case of metronidazole resistance in trichomonosis.
What is trichomonas?
So trichomonas, remember, let's see, which TWiP did we discuss Giardia? Because that was our first flagellate. It was quite a while ago because I believe we did begin with, we did my research organism, Raqueniello, first, but Giardia was one of the early ones that we discussed, and we've discussed other flagellates also, like trypanosomes.
Flagellates?
Yes, they have a flagellum.
These are protozoans, though, right?
They are all protozoans. All the flagellates are protozoans, but not all protozoans are flagellates.
So it's probably after malaria, right?
Might have been. But if the audience...
number 16.
Okay, so number 16 was Giardia, and it was an opening discussion of the flagellates, and I think we continued that through the trypanosomes next.
We did entamoeba next.
Okay.
The trypanosomes were before. We did 2.
All right, so here's another flagellate, protozoan. Here's another flagellate you could link together with Giardia, with the Leishmania, which are also flagellated protozoans, at least in their vectors. And
they're not apicomplexa, though, right?
Oh, no. Those are the malaria. You're just testing me now, Vince. I know. That's okay. I can pass this test. I haven't passed beyond the stage of recognition of the fact that I used to teach all of this.
You said they could be linked.
Linked.
In what way?
No, by their common biology. Okay? They're flagellated organisms. So one must assume, one must presume, rather than assume, that there is some phylogenetic linkage between all of the flagellates. And Giardia happens to be at least one of the most, they call them the most primitive of all flagellated organisms, and maybe all eukaryotic organisms. If you look on the phylogenetic tree as you line it all up, even recent versions of this place Giardia really close to the break between the prokaryotes and the eukaryotes. Mm-hmm. So you're looking at a very primitive group of organisms, the flagellated organisms. And so here's one that's found its way as an obligate extracellular parasite in the urogenital tract of both males and females.
Humans. Other animals?
There's a lot of versions of this infection out there.
Are they different?
They're all different, but they all employ similar biologies, and that is that they're microaerophilic. And when you say that word, Vince, it conjures up a world of the microbiome, right? The microbiome of us and bacteria, because we use these words when we describe obligate anaerobes and obligate aerobes, and then you have this intermediate group that's sort of a microaerophilic group. And that's when I first learned those terms, when I took my microbiology courses, right? and psychrophiles and thermophiles and all these other characteristics. This organism is it tolerates oxygen.
But it can grow without it?
It grows very well.
Because you say it's an anaerobic protozoan here in your book.
I appreciate that. And how old is the book?
Well,
right, 2005. 2005.
Yeah. So we've learned since then that it can survive in the presence of low concentrations of oxygen.
It does, but it doesn't necessarily do its best work under those conditions. So it prefers an anaerobic growth condition.
And where would it find that?
Well, it's below the mucus layer, along the epithelial lining of the urogenital tract, and that's where it lives.
It's above the basement membrane.
That's correct.
Okay.
It doesn't exactly invade cells, right? But it lives in a very specific region of the urogenital tract.
Only the urogenital tract, nowhere else in the
That's
body.
correct. If you swallowed it, forget it.
Even immunosuppressed people, just urogenital tract, like AIDS patients, do they have problems with this going elsewhere?
Not that I'm aware of. I've never heard of this invading. There are some examples of organisms that do that, by the way, but this is not one of those. As far as I know, this is restricted to the urogenital tract.
So there are no reservoir hosts, right?
No.
Just people to people.
That's correct.
And how does it get transmitted? Sexually?
Sure.
That's it? No other way?
That's right.
Okay.
As far as we know. As far as we know.
Can you, if the mother is infected, can the baby get it?
Well, there's another caveat here, too, and that is that it has no cyst stage, okay? So a lot of these protozoan infections have cysts. They round up, and then they can resist the environment because they have to go out in the environment to get to the next person, to the next host. Like Giardia, for instance. Giardia is one of those flagellated protozoans, lives in the gut tract, it rounds up, the nuclei divide, so there are now 4 nuclei per cyst. The cyst goes out in the environment. It's environmentally resistant. You swallow it. It hatches out into 2 trophozoites, and it begins its life again in your gut tract. This protozoan has no known cyst stage whatsoever.
It's always a trophozoite.
It's always the flagellated trophozoite.
Yeah, see, in your life cycle, it's the same.
Yeah. We've never found another one. And in years of culturing this and looking at all of its metabolic states, both aerophilic and anaerobic, it's never rounded up and formed a cyst to say, this is the missing link between how it gets from one person to the other. It actually is transmitted by biological fluids, which incorporates this into that.
So it's transmitted by sexual activity. It reproduces and inhabits the reproductive tract of either the man or the woman.
That's right.
And how long would this infection forever persist, or is it cleared, or?
That's a great question, because that's the crux of knowing whether this is a temporary pathogen or something that we have to put up with as a chronic infection. And in males, let's just talk about that first, because males are usually the silent partner in this one. Males that acquire this infection can maintain this infection for years without knowing they have it.
No, asymptomatic.
Yeah,
and they can spread it to many people. Well,
this is
depending on their promiscuity. Or
this is
not,
correct.
Right?
This is absolutely correct.
But so males are often asymptomatic. What about females?
Yes, but females are not often asymptomatic. Females are, for the most part, symptomatic, and it depends on the heaviness of the infection as to the consequences of the pathology.
What kind of symptoms?
Vaginal discharge, foul-smelling vaginal discharge. Why? Because this is, at its height of anaerobic metabolism, it's producing lots of intermediate products.
Including putrescine.
Correct. And molecular hydrogen. So here's an interesting organism. If you could harness the organelle inside the organism called the hydrogenosome, which we're going to get to, as a separate unit.
The hydrogenosome. Isn't that Christian de Duve?
Yes, of course.
He discovered it, didn't he?
No. Miklos Müller discovered the hydrogenosome working in Christian de Duve's laboratory.
In Trichomonas.
Yeah, that's right.
Are there other parasites or protozoans that have a hydrogenosome?
There are other related trichomonads.
Giardia?
No.
Which is trichomonads.
Yeah.
It's a unique organelle.
Yeah.
What is a hydrogenosome? Does it have a structure in the cell? Is it particular, like a mitochondria?
It can. Turn the page and you can see them.
Well, I'd like you to say it.
It's a membrane-bound organelle that contains
look at that.
That contains anaerobic respiratory enzymes. which allows it to eventually secrete molecular hydrogen. How useful would that be, Vince, if we could harness the hydrogenosome generating mechanism to produce hydrogen at will?
What do we need hydrogen for?
Vince, come on. I know you're leading me into this because I know you know the answer already. If you had a car, for instance, that had a fuel cell, a hydrogen fuel cell, then you could use the hydrogenosome to manufacture your hydrogen and then combine it with oxygen. You've got your energy, you produce water, and as the result, you've got unlimited supplies because hydrogen is the most abundant element in the universe. So,
Car run by trichomonas.
Trichomonads, or at least a derivative of trichomonads. I'm not saying to do that, but I'm just saying that I guess Craig Ventnor spent a lot of money and a lot of time looking for microbes that will secrete molecular hydrogen, and here's one right under his nose. Not figuratively speaking, or literally speaking, I should say.
So are people trying to
I don't
harvest
know.
this?
I don't know, but I'm struck.
Look it up.
I'm struck by the availability of this metabolic pathway to exploration by industry. So if you want to know what makes trichomonads unique, I would say that the hydrogenosome is the biggest reason for considering them as a wonderfully unique group of
Apparently,
organisms.
it's also hydrogenosomes are also found in anaerobic ciliates,
Okay, so we've
and
expanded their world.
fungi.
Really?
How about that?
Okay, so I've learned
The
something
best
here.
studied is that of trichomonads.
Right. And if you go back to the original reference, it's Dr. Miklos Miller.
Can you recite the entire anaerobic cycle, Dickson?
Certainly not.
Okay.
That's not my fort. No, I don't do things
Got
like that.
the acetyl-CoA in here.
I looked them up. I
1 micrometer in diameter.
yeah. Yeah, they're small. And, you can look at these forever and not know what they are. And or you can apply cellular biology to them and isolate them individually and then find out.
Like mitochondria, they're bound by distinct double membranes.
Bingo.
Wow.
There they are. I mean, you can see them right there in the picture, Vince.
So do they have a genome? Yes.
Good question.
No, it's lost the genome. They may have evolved from mitochondria by the loss of
Right.
features, including the genome. So the hydrogenosomal genome is not detected in Trichomonas.
No.
But it is in cockroaches.
Nor do they have mitochondria.
The ciliate, there's a ciliate of cockroaches. Did you know that?
Yes,
It's called
aciliate.
Nyctothyrus ovalis, and there is a genome in the hydrogenosome of that
Wow.
ciliate.
So maybe that's the origin of this. Who knows?
Very interesting.
Yeah, fascinating. Fascinating. But it's not fascinating if you have it, of course, because then there's some pathology associated with all this.
How many people have it? Do you have any idea?
It's a large number, Vince. It's a big number. Like, I don't know if the exact surveys are out from various countries, but I do know that trichomonads are implicated, along with other sexually transmitted bacteria, in creating a lesion in the urogenital tract, which then makes those individuals more susceptible to HIV. Because, as you know, HIV has to be transferred in a living cell.
Yeah.
And
right.
here's the living cell, and now here's a point of entry. So that there appears to be, epidemiologically, a link between the presence of Trichomonas and other bacterial infections like Neisseria and the susceptibility to HIV infection.
So apparently there are other species other than T. vaginalis
Oh, yes.
that are found in humans, too, in immunocompromised patients.
Okay. So,
But what I was looking for
yes.
was the incidence.
Uh-huh.
Worldwide, the annual incidence is about 170 million cases.
That's an enormous
But
cases.
it's not life-threatening,
Cases.
right? It's
These
just...
are these clinical cases?
Because talking about the case fatality.
That's no, not the case fatality rate, but if this is the case rate, then the answer to the number of people actually infected with this is much, much larger.
About 1 in 150 women develop Trichomonas vaginalis, vaginitis. So this is considered a sexually transmitted disease, right?
Absolutely.
So if you have it and you are uncomfortable, what do?
You go to your doctor, obviously, and then
How do they diagnose it?
Well, they can do it several ways. I mean, I think there's a PCR test for this now. They can Obviously,
Wow, it was even in your book, PCR.
sure. But I think it's a standard test now,
Yeah.
by the way. Or you can take a swab sample and you can examine that under the microscope in your office and see them moving around. They're easily cultured, by the way. So,
It says here, if you don't have PCR, anaerobic culture
right,
in medium is useful.
that's right.
And it includes, it's very easy to tell it hasn't, well, actually, based on other symptoms, you can tell an odoriferous exudate, frothy appearance to the
Right.
cervix due to hydrogen plus carbon dioxide. And then they say it's easy to grow out from that.
It is.
And then if they grow it out or if they PCR it, what do you get? Metronidazole?
Metronidazole, yes, which is a known inhibitor of anaerobic metabolism. And that's why it's also useful against, let's say, brain abscesses, which are caused by bacteria. This is a useful drug in that sense if you can get it across the blood-brain barrier.
It's also called flagell, right?
It's also called flagell. Why do you think they call it flagell, Vince?
Because this has a flagellum.
No, because it's effective against organisms with flagella.
Is that right?
Yeah. Like-
So it's broader than just Trichomonas.
That's true. It will defeat, let's say, Giardia. But you can see how rapidly, in some cases at least, a resistance to that drug has developed. The drug was being used in Europe for many years before it was approved in this country because it has some interesting side effects for some anaerobic bacteria that produce carcinogenic-like molecules. And the Ames assay actually was used to implicate metronidazole in certain epidemiologic situations where cancers were more frequent, and said, well, this produces a known carcinogen as it degrades the metabolic pathway through interference with its anaerobic acceptor enzyme molecules. This was completely dismissed eventually because those molecules were then in turn modified further, rendering them non-carcinogenic inside the organism before the organism actually died. So Dr. Mueller was actually instrumental in showing some of those metabolic pathways and diffusing the issue with regards to the use of metronidazole. But like I said, one of our residents here at Columbia Presbyterian Hospital uncovered the first metronidazole-resistant case of Trichomonas, which I then brought to him through a thioglycolate culture, which is an anaerobic culture,
Right.
and Miklos then grew it out and showed what the resistance was due to.
What does it do to?
I forgot. Sorry, I didn't carry the experiments in my head that far.
Do you think we know now?
I think we might know what the mechanism for resistance is. It's probably well known because all of the enzymes, by the way, have been cloned and sequenced from the hydrogenosome. So we know what all of the pathways
So if you are being treated with metronidazole for Trichomonas and it doesn't respond, what do you do?
Well, then tinidazole is another drug which is used. It's an antifungal agent, and some of you may recognize it as an athlete's foot treatment.
That's right. In most cases, metronidazole-resistant isolates remain susceptible to tinidazole.
Yep,
But cross-resistance can be a problem.
it can. And then what do you do? So you try to alter the pH of the vagina, the vaginal area, to make life uncomfortable for the protozoan. All right, so vinegar douches, they use those. It's a mild acetic acid, and the organism doesn't do well under those circumstances. And so maybe you can kill it off simply by doing frequent vinegar douches, but I think that women have a very difficult time getting rid of this infection. They tend to reacquire it, especially if their husband has it and they're not aware of it. So he might be the silent partner, is what
Right.
we used to say here. So you can reacquire it because there's very little...
Well, if that happens, I would think that you have to check the partner to make sure you're not going to get reinfected, right?
Sure, but
treat them.
The problem is that most men, don't like people massaging their prostates to make sure that they can get some fluid to examine to see whether they've got this infection. So I don't know exactly how many male diagnostic tests are run every year. And you could look in other ways too. You could probably do a PCR and seminal fluid or something like this if the male partner were willing to subject themselves to it. But for the most part, it's not a life-threatening infection.
Right. It's just annoying, so people don't want it.
It can go beyond annoying sometimes. It can actually be disabling. People can be bedridden for some time for this and become depressed, and it affects all of the other aspects of your life once your sex life is upset. So that has some sociological consequences as well.
So when you worked here at Columbia Presbyterian in the parasitology lab, did you see Trichomonas frequently?
No. It was never diagnosed in that lab. It was always through OBGYN. Always. And it's always done on, the original diagnosis was done on just examination, and the odor was a dead giveaway.
Right.
But because diagnosis is now easier, they can use other tests.
Cool.
Yeah, and it's all over the world. I mean, this is a universally distributed parasite. And there are different strains, different pathologies, a whole bunch of things that are known about this now that in the beginning, when this emerged, we were unaware of it. But I'm intrigued by the use of the hydrogenosome for peaceful uses, so to speak.
Yes, you've talked about it quite a bit, I remember.
Yeah. I'm amazed that no one's picked it up yet.
Does anyone work on Trichomonas in the research lab?
Well, I knew them all because I was in Miklasz's lab, and so I met all these people as they would come through, just as I meet a lot of virologists as a result of being associated with your group now. But I don't know who the current stars are that work on Trichomonas, but I know that it's still being worked on because it's still a serious medical problem. Yeah.
All right. Well, at one time in my career,
Tell us what you did, Vince.
Trichomonas.
You did.
After I graduated from college in 1974, I spent a year working in a company in New Jersey, a pharmaceutical company called Schmid Laboratories.
Schmid. Never heard of them.
So their products included condoms and spermicides. And they made a drug for Trichomonas infections.
Yes.
I don't remember. I've been looking it up here, and it's not clear what it is. I don't know if it was Flagyl or something else. But what we would do would be they would produce lots of the compound, the drug, whatever it was. I think it was an ointment. All right? So Flagyl is taken orally.
Yes.
But this was an ointment, so maybe they were experimenting with topical treatments. And so we would get lots of the ointment and test them for potency. I was in the quality control division.
Got it.
Someday you can ask me about all the other things that we quality controlled in that company. And we did the Trichomonas. We would have a culture of Trichomonas, which we'd grow in broth.
Right.
And we would make dilutions of we would dissolve it in something that would dissolve the gel, and then mix it with the culture and just look at motility, the inhibition of motility. Now, you said the drug hits the flagellum, the motor?
No. It
just hits flagellated.
It interferes with anaerobic metabolism.
It's the metabolism, yeah.
That's right.
Well, I don't know if this was the same drug, but anyway, we just assayed its activity on cultures. And I remember I would look at these Trichomonas almost every day. I'd see them in the microscope. I see these little guys, swimming around. Trophozoites with flagella moving. And we'd mix it, and when they stopped moving, it was... and then we'd find the dilution and compare it. Because they wanted to know when they made a lot, was it active before they would put it in clinical trials or whatever. Yes. So that's I remember it very vividly.
Interesting.
I also used to do other things. We'll talk about that another time.
Sure.
It was a very interesting experience, but I realized after 8 months that I didn't know a thing, and I needed to go back to school. And that's why I started applying to graduate schools.
Good.
Trichomonas.
The world is a better place because you did, Vince.
No,
it is. I do.
Anything else about Trichomonas?
In cattle, Trichomonas is the other name for this disease in cattle. It causes abortion. So it's got some economic consequences as well. So there's a huge interest in the cattle industry to try to keep this out.
What is the effect in cattle?
It causes the abortion of
Sorry, you said it.
calves. Yeah.
All right.
Yeah.
So does that happen in people?
That's an interesting question. I don't think it becomes that intimately involved to prevent that, but I think it creates a painful situation with regards to just having intercourse. So I think that you're not likely to become pregnant during the time that you're infected.
No, you can imagine infection happening after being
You could.
pregnant. I don't
You
know.
could.
But you don't know of any abortion associated.
Well, I mean, I wouldn't say no, but it's very difficult to prove because I don't know if the reader or the listenership is aware of this, but I was told by a very highly qualified gynecologist that of all of the pregnancies that occur during a woman's life, the first one is about 25% spontaneously aborted. So about 25% of all the women who become pregnant first, for the first time, actually spontaneously abort their embryos. So it's difficult to know why that is. I guess it's almost like it's an autoimmune disease that you're trying to get rid of, and that somehow it develops tolerance for, and then the next pregnancy is much easier and it implants, and then you accept it. Whereas the first one is not readily accepted. And whether this organism plays a role in that or not, I don't actually know the answer. But maybe we can hear from our listeners, and maybe someone has insight into this aspect.
How about we do some email?
Okay.
It's a joke. We did it already.
Well, we could do more. No, really.
Well, we've done this for now an hour and 10 minutes. That's enough. We can stop.
Fair enough.
Keep the email for next time.
Yes.
And let's think about next episode.
No, the next episode will be these free-living amoebae, which we want to talk about.
Okay.
We want to talk about Balmuthia mendrillelaris,
Niglaria.
and Niglaria fowleri, and
Castellani.
Castellani.
All
We want
right.
to do those three next time.
That would be fun. You know, there are periodically in the news people who die of
They are.
Niglaria fowleri. Right.
This is true.
All right. That'll be next. You can find TWiP. We don't have to find it because we do it.
That's
You
right.
can find TWiP on iTunes, at the Zune Marketplace, and at microbeworld.org/TWiP, where we keep all the show notes. If you do like TWiP, go over to iTunes and leave a brief comment. It helps us to stay visible and get more listeners. And do send us your questions and comments to twip@twiv.tv. Dickson Despommier can be found at Raqueniello.org, verticalfarm.com, and medicalecology.com. Thank you, Dickson.
You're welcome, Vincent.
I guess you're hungry now. You'd like to have
Pleasure.
some lunch, right?
No? Not necessarily.
Well, I have my lunch. Do you want to eat lunch? Or are you going to have lunch with someone?
I'm not going to have lunch with someone. I'm going to have lunch with you.
Right. All right. Do you have to go buy it?
I do.
All right. You can go do that. I'm Vincent Raqueniello. You can find me at virology.ws. You've been listening to This Week in Parasitism. Thanks for joining us. We'll be back soon. Another TWiP is parasitic.